Hello, everyone, and thank you so much for your interest in this project and for taking the time to view this presentation. My name is Sever Siberian, and I'm a fourth-year medical student at Morehouse School of Medicine. In this presentation, I'm going to share a brief history of the flow of genetic information, the new discoveries from our lab, and the implications of these findings in developing new therapeutic agents. We have no conflicts of interest to disclose at this time, and the sources of funding for this project are also listed below. Here is a snapshot of our poster, which will be available as a separate file. And to start the introduction, I would just like to visit the central dogma of biology, which is the flow of genetic information from DNA to RNA to eventual translation via ribosomes into proteins. There is also non-coding RNA category where microRNA, small nucleolar RNA, and long non-coding RNAs exist. And these RNAs do not code for any proteins. And because of that, they have always been referred to as junk DNA and RNA in many textbooks. And in a lot of resources, you'll see that 1.5 to 2.5% of genomes code for proteins. However, what if non-coding RNAs were able to produce proteins? Can we still say that this is considered junk DNA or junk RNA? In terms of the synthesis for non-coding RNAs, the simple mechanism is that you have the microRNA gene getting transcribed into primary microRNA or miRNA, which then gets further processed into pre-miRNA, and eventually it is transformed into a mature miRNA, which has its functional abilities. We're going to build on this model based on the data to talk about the significance. A novel pathway that I'd like to share with you now is this new step in this process that we have discovered in the lab. It is a production of a protein from the miRNA stage, which actually shows us that the non-coding RNA actually does code or can code for proteins. And this was proven in our lab by finding an open reading frame in miRNA, which was then translated into a protein via ribosomes. What is the significance of this? Well, what if the miPEP itself is able to have some sort of functionality? We're going to take aortic medial calcification as a model to show this. Aortic medial calcification is a disease that has a well-established pathophysiology, and we know that it's due to hypercalcemia. Many different things can cause hypercalcemia, and I've listed a few of them here. Hyperparathyroidism is the one that we focused on for our literature search due to its involvement with pKa. We can measure osteoblastic activity by way of biomarkers such as alkaline phosphatase in the bloodstream to quantify the amount of calcification in the tunica media of the vasculature. And you can see the clinical progression of the disease at the bottom as well. To build the model that we were just talking about, we can actually add these few extra steps where miPEP has potential activity that could either stimulate the production of mature miRNA, or it can decrease the activity of the product of another gene. In terms of aortic medial calcification specifically, we can see that miRNA getting translated into miPEP-200b, here it can actually act as an inhibitor of the pKa enzyme, or it can stimulate the production of mature miRNA, which in turn inhibits pKa mRNA and its functionality. In either case, you have decreased pKa enzyme activity, decreased PTH or parathyroid hormone, and decreased downstream sequelae of this process. And some of the research that backs up this hypothesis and this proposed mechanism, they are all listed on the side here for your reference. And so why is this very important to us? Well, we have to now revisit previously found mechanisms of action, because what if miPEP is involved and we always thought that it was miRNA? We can also take miPEP as a great marker because it has much greater stability in the bloodstream than do RNAs. And we can also now reconsider the term junk when talking about these DNAs. Most importantly, though, it allows us to have additional sites of targeted therapy. Whereas before we had only one site, now we can target miPEP, we can target the miRNA, we can target pKa. And so lastly, I want to talk about the impact of COVID. With the advent of this new RNA vaccine that came out of the pandemic, combined with our recent findings, we're planning to look into the possibility of using the technology for treating diseases where miRNA is classically the culprit. RNA does not survive in the bloodstream because of its instability, so if we can give RNA vaccine that allows for production of miPEP in myocytes and then releases it into the circulation, we may be able to treat diseases that are deemed impossible to cure. And so we are truly at an incredible time in medicine and pharmacology with so much potential for future advancements. Lastly, I'd like to acknowledge the lab members of Dr. Reddy and Dr. Rao's laboratories, and also thank Dr. Reddy for his mentorship and for teaching me everything about his craft. These are our references, and thank you again for your interest in this project.